All right. Good morning, ladies and gentlemen. Thank you for joining GenScript webinar today. For your information, we will be discussing the advantages of cyclic peptides and their current market status and their key pharmaceutical applications in drug development process. Allow me to introduce our speaker today, Dr. Mina Wang. She is the senior peptide scientist in GenScript. And before joining GenScript, she earned her PhD in chemistry from the University of South Florida. Her work in GenScript has been focused on establishing and updating the cyclic peptide service for various applications. Before we get started, here are a few housekeeping notes. We have a dedicated Q&A session at the end of this webinar, so feel free to leave your question at the Q&A box and we will address to them later. This webinar is being recorded and the recording will be made available to all participants afterwards. Without further ado, I'll pass the stage to Dr. Wang. Dr. Wang, over to you. You can unmute your mic now.
Okay.
Yeah, just wait for
I
a
guess
while. Yeah, just wait a while for Dr. Wang to reshare her slides.
okay. Is everything good?
Yeah, it's perfect now. All right, over to you.
Okay. Okay, guys. Hello, everyone. Good morning. And I am Mina, a peptide senior scientist in GenScript, the peptide department, and I'm responsible for the cyclic peptide synthesis update. It's quite exciting to have this opportunity to share our experience and have discussion on cyclic peptide with you. So let's get started. I will cover four parts today, including the advantage of cyclic peptides in drug discovery, an application of cyclic peptides, and synthesis of common cyclic peptides. Last but not least, case studies of cyclic peptides in GenScript. Since this presentation would be a little bit long, so if you have any questions, you can interrupt me directly. Mm, Globally, 23 cyclic peptide drugs have already been approved since 2001, and the total sales of in the last year is around $6,000 million. And many candidates are also in clinical trials. So you may wonder why more and more companies and researchers being attracted by the cyclic peptides. I think we can start with the looking at the advantages of the cyclic peptides. Cyclic peptides have improved the protein-protein interaction ability because they have large interaction surface, and that will make it sufficient size and balanced conformational flexibility and rigidity for binding to the flat protein-protein interaction surfaces. And that kind of binding usually has the antibody-like affinity and specificity. The constrained structures introduced by the cyclization also make the peptide chain more conformationally stable. That will make it that will make it has increased proteolytic resistance. And also the resistance of the linear peptide being extended, being increased by the cyclization. Therefore, their half-life in the blood will also be increased. Cyclic peptides really have uh, longer half-life in the body. And many literatures have disclosed this effect with a lot of real data. And for cell membrane permeability, the peptide cyclization increases the both passive diffusion and the active transport because compared to linear peptides, they have a lower energy barrier for adapting to the membrane environment, and their binding affinity to transporting proteins are also enhanced. So they really have better cell membrane permeability. And except these advantages when compared with the large biodrug cyclic peptides usually have 500 to 3,000 molecular weight. That makes it easy to be synthesized and modified. So usually the production cost is lower. So, what are the most important research fields of the cyclic peptides? I will give a brief introduction about that. We all see that the successful drug development of ADC drugs in global market, PDC drugs, which represent peptide drug conjugation. It is being researched increasingly in these years. The concept of ADC and PDC are very similar. They all consist of targeting parts, linker, and some drug payloads. So compared with ADC drugs, the PDC use the peptides as targeting parts, while ADC use antibody. So PDC has a lot of advantages compared to ADC, such as the smaller molecular weight, better tumor penetration, easier kidney clearance, lower production costs, and a wider variety of drug loading. And due to the good targeting properties and the cell permeability of cyclic peptide, many candidates employed cyclic peptides in PDC drug development. I put an example here. This is approved PDC drugs. It is Lutathera. Um, Lutathera has cells. We can find that the trend of the Lutathera cells increased sharply and then reached to a horizontal line. That means we can see that the promising trend of PDC drugs. However, it also has some limitations, especially for PDC drugs, because PDC drugs has very strict transportation policy and using policies. So we can see there is a top at here. Except the already approved drugs, we also have a lot of PDC candidates in the clinical trials. And in the bicycle therapeutics, it has a lot of cyclic peptide involved PDC drugs. We hope that in the future it will have a very good behavior in the clinical treatment. So next application field of cyclic peptide is CPP, which represents cell penetrating peptides. So this kind of peptides it comes from the application like cell penetrating, uh, because a lot of some peptides or drugs they cannot enter the cell membranes. If we connected the candidates with a cell permeable peptide, the total cell penetrating ability will increase a lot. Um, and as I mentioned before, the peptide cyclization increases both the passive diffusion and active transport. So therefore, it usually has better cell permeability than linear counterparts. So the published data at here can also confirm that. So in this research, we can see that peptide 6 has tenfold better cell penetration ability than the peptide 7. So that the result of peptide 11 and 11 and 12. Mm, that's because of the cyclic peptides advantages as I introduced before. To produce this kind of CPP, the application of it is quite simple. Sometimes you can directly fuse the CPP with the peptide targeting parts together, and it can code endocyclic delivery. And also we can conjugate the CPP part with the peptide targeting part by using the scaffold, the center scaffold, to form the bicyclic delivery method. The scaffold in the center can be changed to many other kinds of structures, and this one has larger interaction surface with proteins. So it also has very promising applications. And if we conjugated the CPP, cyclic CPP, with the targeting part directly, it can be called endocyclic delivery. So as I introduced before, the cyclic peptides have a large interacting surface. So it is quite promising to develop cyclic peptides as protein-protein interaction interference. So globally, many pharmaceutical companies design cyclic peptide library to screen potential hits such as bicycle therapeutics, as I mentioned, and they use phage display and chemical modification to generate bicycle libraries. But here I used an example from a Merck company. They obtained a thiol ether bridged peptide library using the mRNA display and the SN2 chemical reaction to form this cyclic peptide library. And then after several rounds of screening, they got the potential hits, and the hits can be then modified by the SAR, which means structure activity relationship, to make some modifications in this procedure. Usually, hundreds of structures may be designed to get the most promising candidates. This one is MK06 16. This one is in the clinical third in the phase 3 clinical trials to treat the LDL for the as a form of oral pill. So I guess you may come across the situation that the synthesis of your cyclic peptides are difficult. So I'm going to briefly introduce the synthesis of some common cyclization strategies. So the disulfide bond is the most common cyclization strategy according to the statistics. We can see during these 20 years, 7 drugs which has already improved had the cyclization strategy of disulfide. So it is quite promising. It can has 1 disulfide bond, two disulfide bond, or 3 disulfide bonds. So usually, how do we get these kinds of structures? The most common methods are using oxidant reaction, like air oxidation, hydrogen peroxide oxidation, dimethyl oxidation, and iodine oxidation. The air oxidation is usually being oxidized by the oxygen in the air. The advantage of it is is a reaction condition which is very mild. It will it will not be it will not have a lot of side reactions during this procedure, but it also has drawbacks because the oxidization ability of oxygen in the air is not very strong, so it will take a long time to finish this reaction. And if we want to do this reaction, the peptide should be soluble in the water, so sometimes it's also challenging for that. And for the hydrogen dioxide oxidation, it is very fast because dioxide is strong oxidant reagent, but it also has drawbacks like the same as the air oxidation, it has to be water soluble. And the other one is it has side reactions. A very high ratio of peptides will have this kind of side reactions because the strong oxidation ability of hydrogen peroxide. And the DMSO oxidation is much better than these two because it don't have to be water soluble because DMSO can increase the solubility of the peptides in the solution. So it has a broader application field, but however, it's also very slow. Its oxidation ability is not very strong. It's not as strong as the hydrogen peroxide. The last one is iodine oxidation. This kind of reaction usually being applied in the synthesis of the peptides with two disulfide bonds, because the first disulfide bond usually formed by these three methods, and the second one, it is usually protected. The thiol groups in this structure is usually protected by the ACM, and the iodine can cleave the ACM protection group and form the disulfide bond in one pot. So it is fast and very convenient, and the reaction is also cheap. But um, the drawbacks is quite similar as the hydrogen peroxide. It's the reaction, um, the the ability of the oxidation is very strong, and sometimes it will have the side reactions. And also we find some side reactions like reshuffling during the reaction, because the disulfide bond sometimes the location will be changed during this reaction. So to solve these kind of problems, we developed two methods. The first one is cell activation mechanism. We use this kind of reactions to make sure the whole procedure fast, easy, and few side reaction, and it can have mass production. Mm, and the other one is mild condition for ACM cleavage. As I mentioned, the iodine can cleave the ACM group and form the disulfide bond. However, it has side reactions. And here we developed the mild condition for ACM cleavage. Using this condition, it will very it will the ACM protection group will be cleaved in a mild condition. We don't have to use oxidant reagent in this reaction. And if in your structure, if there are some very sensitive amino acids like methionine, tryptophan, we can use this kind of method. And I will give some case studies about these two methods in the later slides. So the lactam cyclization is another one, which is very common in the drug discovery. It can be synthesized on resin or in solution. On resin synthesis steps are drawn here. The only the sequences have asparagine, glutamine, asparic acid, or glutamic acid can use on resin synthesis. I mean, one of these four amino acids exists in the sequences is enough. That's because these four amino acids has side chains which can be connected to the resin, and the hydroxyl groups on the main chain could be used to do the cyclization. So in these steps, we can see firstly the side chain of these amino acids can be connected to the resin, and the main chain of the amino acids can be protected by the OALL protection group. This one, after it's being cleaved by some reagents, the carboxylic acid group can be used to connect it with the uh, primary amine at the end of the sequence. Use some conjugation reagent like HATO or Oxima DIC, this kind of reagents. And then after the forming of the cyclization, we can use TFA to cleave the whole sequence, and then we can get the desired product. But however, this kind of reactions sometimes is not very efficient. I mean, it will work sometimes, but sometimes it will not work. We cannot promise it will work for all sequences. That is a main drawback of this kind of cyclization of resin. And except this method, we can also do the cyclization in solution. The synthesis is quite simple, but it's more complex than the unresin cyclization, because we need to fully we need to get the fully protected amino acid, and then do the cyclization in the solution by this conjugation reagent, and then we need to use TFA to cleave all the protection groups on the sequences. So it's so in general it's a little bit complex than the unresin reaction. Sometimes these two reactions can used to cyclize the sequences with five and with sequences more than five amino acid and less than 40 amino acids. If you have sequences shorter than that or longer than that, it can it cannot be as efficient uh, as as that one. In that situation, we will use NCL cyclization. The NCL cyclization is normally used to synthesize the long peptides, which has amino acids more than 100. It is very efficient, so we can also apply it in the cyclization. It will so if you have sequences less than 5 amino acids or longer than 40 amino acids, it can be cyclized using this method. But it also has drawbacks because this reaction is quite complex. We need to activate the carboxylic acid groups firstly and then do the cyclization. But at least it can solve some problems. So the last synthesis of cyclization I'd like to introduce here is the Cys-Cys bridge synthesis. As I mentioned before, no matter in mRNA display or phage display, the Cys-Cys bridge are used most commonly because this reaction is very efficient and the reaction condition is quite simple. It just needs a mild base condition so that DNA or the reaction can be performed both on resin or in solution, and we have a lot of experience on this kind of synthesis. And there will be I will give a case study about this synthesis after a while. So that's the part is a case study. I will have 4 or 5 case studies. So to help you to understand the cyclization strategy I just mentioned. The first one is a mild ACM cleavage method we developed. For the peptides with 2 disulfide bonds, we usually get it by 2 steps of oxidation. The first one is performed by air, DMSO, or hydrogen peroxide. I just introduced. But for the second one, the ACM protection group and iodine oxidation is almost the only choice. It is fast, clean, and economic. However, nearly 30% of sequences have side reactions during this step. The picture one, I give 2 examples here. The picture one is HPLC result after being treated with iodine. We can see that the disaster have already made by this reaction. Because before the reaction, it's very pure, but after the reaction, it's very it's the impurities is a lot. And for the second example is the reshuffling of the disulfide bond. It looks like a single picket here, but when we zoom in, it's 2 peaks, and the molecular weight of these 2 peaks are the same, which means the disulfide bond has already been changed. It already have 2 products here, and we can see the retention time of these 2 peaks are very similar. So it's impossible to separate these 2 peaks. To solve these 2 side reactions, we developed complex reagents which contains reagent X and reagent Y to solve this problem. The reagent X is to grab off the ACM group, and the reagent Y is to form the disulfide bond. The reaction is a one-pot reaction, so it's very convenient. We just need to add another reagent after the grab of the ACM groups. And for the reagent Y, we do not use oxidant reagent because oxidant reagent sometimes will give us the side reactions. We use the thiol activation mechanism reagent to form the disulfide bond here. And since this is a one-part reaction and we have less side reactions, the yield of of this reaction is really higher. So this reaction has already been broadly used in our department, especially for some sequences which is very long or with some sensitive amino acids like methionine or tryptophan. So the second case study is about the new disulfide formation method. Recently, to meet the demand of the market, we launched our cyclic peptide library service. The capability of providing cyclic peptides with one disulfide bond has already been increased to 1,500 to 2,000 per mass. Price and turnaround time were also updated. You may wonder how can we do that? How did what kind of method we have used to realize this kind of capability? So the new disulfide bond formation method and peptide library platform are the two keys that guaranteed our cyclic peptide library synthesis service. The new method has the mechanism cell activation instead of the oxidation. Actually, to be honest, the method of ICM, the protection method I just mentioned in the last case study, also used this reagent as reagent Y. We can see here the reagent Y here is actually the new method we have developed. So this new method has a very short reaction time. We usually can finish it like in 5 or 10 minutes, and the yield is very high. The general yield is around 40% to 80%, and it can be performed as a mass production. I mean, we can do it in a very large amount of peptides at the same time. And also for the peptides library platform, we can produce 600 peptide per batch in 24 hours. So it is a I can say that it is a world biggest peptide production platform. And this picture is HPLC, a result of a new method. We can see the linear peptide has a very good single peak, and the crude product of cyclization also has a very similar HPLC result, which means it has no extra side reactions during this reaction. And the third case study is about a new cell protection group. Basically, it is a case study about how to protect cell groups orthogonally. In some sequences, there is another free cell group in the cyclic peptides with disulfide bonds or cell ether bonds. So this cell group can be used to form a redox sensitive linker or non-cleavable linker in PDC candidates. And sometimes these cell groups can also be an important binding site for the affinity. The common ways to get this kind of peptides are on resins cyclization strategy or using some protection groups like acetybutyl. However, they're all unsatisfactory solution due to the low successful yield because these reactions are quite sequences dependent. We have applied this kind of method in our production. We found sometimes it will work, but sometimes it cannot work. That's really not good. So we developed a protection group, which name is MX, as a safe way to make these peptides. It can works for almost all peptides, and it's not which means it's not sequence dependent. The MX has no, it is very stable to TFA, so we don't have to worry about the cleavage in the cleavaging of the whole peptides. And the cleaving condition of MX has no influence on the existing disulfide bonds or other bonds. the most importantly, MX has realized internal supply in GenScript peptide department. So we have a very low cost of this kind of peptide sequence. And the GenScript and the HPLC, we can see the HPLC result it here. It is result from these sequences. The MX has already been cleaved. We can see the crude product is quite pure. And during this reaction, we don't have to worry about the disulfide bound reshuffling, and we also don't have to worry about the compatibility of this protection group. It can work on sequences with any kind of amino acids, and it can be cleaved very easily. And so in general, the yield is very good. The fourth case study is about the bicycle PDC peptides. In this project, we have 85 bicycle peptides applying for PDC application. This is a project from a very big farm, and in this 85 bicycle peptides, we have four kinds of scaffold. The first one is TATA, which is a commercial available structure, and is used very commonly in the PDC drugs. And we have another three scaffold, which provided by our clients. And so in summary, we get the completion ratio is 96.5%. The difficulties we came across during the synthesis of these peptides comes from three parts. The first one is linear peptide synthesis. The second one is cyclization. The third one is purification. For the linear peptide synthesis and purification, to be honest, they come from the hydrophobicity of the peptides. If we know that if we have too much hydrophobic amino acid in the peptides, it will be the synthesis would be very hard. And also, if you have some sensitive amino acids, it will be easily oxidized during the cleavage. To solve these two problems, we updated our synthesis strategies, like switching the conjugation reagent from the commonly oxima DIC to strong reagents like HATO, HCTO, HBTO, this kind of reagent. And for the purification, we changed our columns from the normal C18 column to like C4 or some other kinds of columns. And today I'm going to share some experience about the cyclization difficulties and how we get rid of the difficulties. Mm, so in this reaction, we use the reagent A, reagent B, and linear peptides to get our bicycle peptides. And the most common side reactions come from the side product one and product two. The product one is the insufficient reaction of the scaffold. We can see only two cell groups participate in the reaction, but there is another free cell groups and a free double bonded here. And most importantly, this side product has very similar retention time of the when compared with the product. And that side product is the extra um, reaction of the scaffold. We can see there are two scaffold on one single peptide. So to control the side reaction production, we adjusted the reagent A and the reagent B. The ratio of them is very important. For example, we adjusted to A1 equivalent, A2 equivalent, and A3 equivalent, and we did the comparison on the HPLC, and we find that in the I guess the best one is the A1 when we use A1 equivalent reagent B. So that means we already have the experience of synthesizing these kinds of bicycle peptides, and we have already the whole project has already been delivered successfully. The last case study is about a stable peptide. The stable peptide is a short peptide, typically in a alpha helical conformation. That is, the conformation is constrained by a synthesized brace. It's a double bonded here. So usually stables are synthesized using the ring closing metastasis, which briefly called RCM reaction. The alpha helix are the most common protein secondary structure and play a key role in mediating many protein-protein interactions. Introducing synthetic brace helps to lock the a peptide in a specific conformation, which can reduce the conformational entropies when it interact with the protein interaction. So this approach can increase the target affinity and increase the cell penetration, and the project the protect against the proteolytic degradation also be increased. So in this project, we finished 17 stable peptide with heteroatoms on the bridge. So it's not only consists of carbon on this bridge, we also have some heteroatoms on the bridge. The double bond formed in the product will have Z and E, two isomers. These two isomers have similar retention time and they're difficult to be synthesized. We can see on this picture the E isomer is higher and the isomer is lower, but they have very similar retention time. So we applied another the GRAPS2 catalyst to produce this reaction to have a, a better different to have a higher E ratio in this product, because if you use GRAPS1 catalyst, D and E conformation will have very similar ratio and it makes the separation harder. But if you use GRAPS2, the situation would be better and you can have uh, higher ratio of product E. And how to separate, I mean how to identify these two products. We know that this is product E, but how do you know it is product E? We need to use NMR to do this identification. So we have the two-dimensional NMR to identify the product. If the resolution of the two peaks is larger than 15 hertz, it means this product is E isomer. And if it's less than 12 hertz, it means it is Z isomer. And usually, E isomer has better activity than the Z isomer. So as a summary, GenScript can provide more than 30 kinds of cyclization strategies. The capability is over 2,000 cyclization peptides per month, and for the large-scale synthesis, more than 3 kilogram cyclic peptides can be produced per month. And we also have express synthesis service, which can permit the turnaround time as less as 6 days. Except for the customized cyclopeptide synthesis, we also provide the library synthesis. So now we have head-to-tail cyclic library service and disulfide cyclic peptide library. The first one, we have the capability of produce 750 items per month, and for the second one, we have 2,000 items per month. Not only peptide synthesis, we can also provide one-stop solution service from your drug discovery stage to clinical research period, such as process development, dossier submission, quality control, and CGMP production. The CGMP production is what I want to mention here. So, encouragingly, our new peptide GMP for facility was launched in this year. The 2,000 square meter CGMP facility with C manufacturing suites has two lines, two CGMP manufacturing lines, API production line and the new antigen peptide. The clean suit with grade A isolator in grade C background for stereo filtration and aesthetic fill and finish will be ready in next year. So you are very welcome to visit our sites and discuss with our professional teams. So thank you very much, and that is all what I want to share today. If you have any questions, I will be very happy to take it. Thank you very much.
All right. Thank you, Dr. Wang. I have a couple of questions from the attendees, and I will read them out to you, and I need your help to answer them. And for
Sure.
all the attendees out there, if you have any questions, feel free to leave it in the Q&A box. All right. So the first question is, I'm very interested in the MX protection group you mentioned in the case study. So can it be used in the synthesis of sequences with three disulfide bonds orthogonally?
Thanks for your question. Yes, of course. It can the reaction can be stopped at the MXD protection group, and also it can form the disulfide bond continuously. So we can apply it in the peptide with three disulfide bond synthesis. Yes, yes, we can do that.
All right. Okay. And the next question is, I noticed that you offer a library synthesis service. However, synthesizing numerous peptides can be labor intensive and costly. So, do you have a more efficient method for obtaining a peptide library?
Yes, I have to say that synthesized peptide is very can have accurate and more convenient pull down assays. But if we want to lower the cost and lower the and faster, we can use some other kind of method to get peptide library with a very large amount, like OBOC, a one bead, one compound library, and we can also use mRNA display, phage display. But in our department, we can apply one bead, one compound library synthesis method, which can have the millions of peptides in this library.
Great.
Mm.
Yep. And we had a structure with both free thiol and disulfide bond. It was very unstable, with the reshuffling of the disulfide bond occurring even during the process of lyophilizations. Do you have any suggestions for this issue?
Oh yeah, we also have this kind of issues during our synthesis. Usually we will lower the temperature of the lyophilization. Sometimes it will have very good results. So I guess you can try this method.
Yes. Okay, the next question is, do you have a library of common cyclic peptides for the peptide screening?
Uh, uh, Sorry.
Do you have a library of
Mm-hmm.
common cyclic peptides for the peptide screening?
Oh, yes, yes. For the structures for the cyclization strategy for screening, to be honest, if you have a library, it means there's a lot of peptides in your library. So the cyclization strategy usually is not the main factors affect your cell activity. As long as you fix the linear peptides, that is enough. I will recommend cell ether bridge as your cyclization strategy because this reaction is very simple, very convenient, and very efficient. It can promise that almost all of your sequences in your library can be cyclized. But if you change to other kinds of strategies, it cannot promise that. So cell ether bridge would be a good choice.
All right. By the way, Dr. Wang, you may check the Q&A box. You can unshare this slide so it can help you better to understand the questions. The next question is, how is the prospect of using enzymes for peptide cyclizations compared to chemical synthesis?
Um, to be honest, in our department, we do not have enzyme catalyst for the peptide cyclization, because I also read some papers about the using of enzyme, but I have to say that it is very picky for enzyme cyclization. You have to have several specific amino acids in your sequences to use enzyme, but for the chemical synthesis, it has no limitation. You can try any kind of cyclization you want. So I think the chemical would be broader, and the enzyme. The advantage of enzyme is green chemistry, where it has no harmful reagent, but as I said, it's picky.
All right, I understand. Do you have an experience with epimerization during peptide synthesis?
Epimerization, I think. Um, could you please explain epimerization a little bit for me?
Okay, maybe we'll come back to these questions later. Yeah,
Mm-hmm.
don't worry about it. Yeah.
Okay.
And the next question is, peptides with disulfide bonds are not very stable in cell culture. Do you have any solutions for this issue?
You mean the disulfide bond not very stable?
Yeah, it's unstable. Yeah.
Mm, I think compared to disulfide bond, the the disulfide bond you can change it to cell ether bond. Cell ether bond is much stable than the disulfide bond, and sometimes you can also change it to lactam bond. I think lactam bond is also stable to the reduced atmosphere in the cell culture. Yeah, is that
Oh,
answered your question?
yes. Uh,
I see the last question.
yes. Okay, so please comment on the stability of cyclic, bicyclic, and tricyclic compounds and their synthesized cost.
So, okay. I think the stability of cyclic peptides depends on the bonds which make the cyclization, not the number the number. Um, for example, if you have the three disulfide bonds in your peptides, it is still not as stable as cyclic peptide with the lactam, because the disulfide bond is very easily to be reduced in the cell culture, but the other bonds like stable peptide or lactam peptide, it is much stable. But if you compare the disulfide bond with peptide with one disulfide bond or two disulfide bond or three disulfide bond, obviously the three disulfide bond would be stable than the peptide with only one bond. And for their synthesis cost, definitely the more chemical synthesis steps, it will cost higher. The three disulfide bond cost much higher than one disulfide bond, especially you want the bonds orthogonally. So I think I answered that question. What is the next one?
Yes,
Mm,
I think we have answered most of the questions, and there are a few remaining here that require further investigation and more research in order to
oh,
answer their questions. So we will
okay.
answer them offline, and we'll be sharing all the questions and answers with all the attendees later after this webinar. All right.
Okay. All right. I think I got the question. The uh epimerization. I think I can
Oh,
answer that question. Yeah.
okay, great. Sure, sure.
Okay, okay. So the epimerization during the peptide synthesis sometimes is unavoidable, but you do have some strategies to avoid that. For example, we can use the dipeptide to replace the peptide to replace the peptide being conjugated one by one. For example, if we have the um, G amino acid and C amino acid, sometimes we know it's very easy to um, to epimerization. But if you connected these two amino acids together before you instead of adding them one by one, sometimes it will have a very good result. I think you can try this method. So, how is the perspective of isolopeptide from industrial perspective? I think I will do some research on the isopeptides because I don't think I have experience on these peptides. So
Yeah, don't worry
sorry.
about it. You can, yeah, well, we can do some research and we get back to the
Hmm.
attendees later.
Okay.
Yeah. So I guess this is the last questions, and we have come to the end of this webinar. Thank you so much, Dr. Mina Wang, for your
Hmm.
great presentations. We really appreciate it, and thank you all the attendees for your time in joining our webinar today. If you have any questions or you need more information, please visit our official website www.genscript.com for more information. Thank you so much for your time today, and we will dismiss the webinar now. Thank you.
Thank you all. Thank you for your time.
Yeah.